Hi, Dr. Ross. These are my lab results. For part one, we had to do two graphs by hand. So first one was weight versus height. I plotted the points based on the values given. And this one had a best fit line trying to connect as many points in between as possible. Graph two is observance versus the wavelength. It's a little bit different because it is a smooth curve graph. So this is connecting all the points from the values given. In part two, I inputted the values into Excel. So we had two graphs. First graph is the density versus the percentage of the sucrose. So once I have the graph, I inputted the trend line, which is a dotted line trying to connect as many points as possible. I also included the R squared value and the slope line value. So the R squared is actually greater than 0.90, which means that it's almost a best fit line connecting the dots. In part three, we had to input the values onto an Excel sheet. So this is my results for the values where I had to calculate the density, the average mass, the percentage of density, and then the average of the density and the percentage error for all of the values. And then part four, we had to create another graph. So this is absorbance versus the concentration. And then given the values, I inputted the trend line, got the R squared, the slope line, and then that's where I got the results for the unknown concentration, which is in part five.